Okay, let's start. Thank you for attending the webinar. Arigo would like to update some research trend about the targeting cell death, which is important for cancer therapy. Let me introduce Arigo first. There is a squirrel hidden in our logo. Arigo care about quality. We supply specific and reliable antibody product. And we follow the research frontier and bring out cutting-edge research solutions. Our goal is to offer smart solutions such as antibody duo, antibody panels, and ELISA kit to facilitate scientific research. Let's back to cell death. There are two groups of cell death. One is necrosis, which is accidental cell death due to cell injury. The other is programmed cell death, which is controlled cell death. The best known programmed cell death is apoptosis, which was found half a century ago. In 2001, a new type of cell death was found. That is Ferroptosis. And in 2005, Necroptosis was found. More recently, the Ferroptosis was found in 2012. All these are different types of cell death. Let me talk more about apoptosis. Apoptosis is required for morphogenesis in embryo stage, and in adult, apoptosis is required to clean dangerous or harmful cells. It is a cellular suicide to remove mutated or infected cells. The mutated cells are the source of tumor cells, and the infected cells, for example, the virus-infected cells, proliferate many viruses to infect more normal cells. All these are harmful cells, so these cells have to be removed by apoptosis. If the cellular suicide is failed, the immune cells mediate cytotoxicity to eliminate infected or tumor cells. That means the immune cells kill the infected or tumor cells. Apoptosis works through caspase cascade. That a lot of caspases are involved. Caspases are a group of proteins that can cleave many proteins, including different caspases. There are two pathways involved in apoptosis. One is intrinsic pathway. The cellular stress activate initiator caspases such as caspase 12 and caspase 9. The initiator caspase then activate the executor caspase to induce apoptosis. The other pathway is in extrinsic pathway. The ligand binding of death receptor or TNF receptor activate caspase 8. The caspase 8 then activate the downstream caspase to induce apoptosis. Both intrinsic and extrinsic pathways eventually activate caspase 3, that is the executor caspase. Caspase 3 cleaves many other proteins and result to apoptosis. PRP is one substrate of caspase 3. So the cleaved caspase 3 and the cleaved PRP are recognized as markers to detect apoptosis. So these antibodies are used as apoptosis marker antibodies. Besides, an alternative pathway is the cytotoxicity. the cytotoxicity is mediated by immune cell. The immune cells release granzyme and perforin. Perforin form a pore in the plasma membrane. Then the granzyme enter the target cell to induce to activate caspase 3 and induce apoptosis. So the immune cells induce the target cell undergo the apoptosis process. The cytotoxicity is important for anti-cancer immunity because higher cytotoxicity means the immune cells have higher ability to kill the cancer cells. How to examine the cytotoxicity? The hallmark of cytotoxicity is the elevation of granzyme B and the perforin expression. So the expression level would be increased, and the increased expression could be detected by granzyme B and the perforin antibody by staining. Please keep in mind that cytotoxicity would induce the apoptosis through release the granzyme. Please keep in mind this. I'll talk this later. So, targeting apoptosis is important for cancer therapy. In tumor cells, the apoptosis is inhibited. One reason is the malfunction of BCL2 family member. BCL2 family is a group of protein family. They can be subclasses into there are two subclasses in the BCL2 family. One is the anti-apoptotic family, apoptotic protein that includes the BCL2 and the BCL-XL. This protein can prevent apoptosis, so they have anti-apoptotic role. The other subclasses is apoptotic protein that include the BAX and the BAT protein. This protein induce apoptosis, so this protein trigger the apoptosis. In tumor cells, many anti-apoptotic proteins are overexpressed. The BCL2 and the BCL-XL are overexpressed, so the tumor cell, the apoptosis in the tumor cells is failed, so the tumor cells survive. So the strategy for the malfunction of BCL2 family is to develop the inhibitor of the anti-apoptotic protein. Here show you the update about the on developing inhibitor. You can see this one is dual inhibitor that inhibit both BCL2 and the BCL-XL, and this three is BCL2 inhibitor. This four is BCL-XL inhibitor. All these inhibitors can be used for cancer therapy, but all these inhibitors are still in very early phase of clinical development. You can see most are in very early phase, such as the preclinical phase or phase 1 or phase 2. Then let's talk about the necroptosis. Necroptosis is a backup of apoptosis. In tumor cells or infected cells, the caspases are inhibited, so the cells, the harmful cell, the apoptosis is failed in the harmful cell. In this case, the cells have alternative mechanism to to clean the tumor cells or infected cell. That is necroptosis. The phosphorylation of three proteins are important for necroptosis. The first one is RIP1 phosphorylation. The phospho-RIP1, it is a kinase. The phospho-RIP1 then phosphorylate the downstream substrate, that is RIP3. Then the RIP3 phosphorylate its downstream substrate, MLKL. The phospho-MLKL protein has pore-forming ability. So the phospho-MLKL protein form a pore in the plasma membrane and result to plasma membrane broken. So there is a hole. The plasma membrane is broken. Then the cell die. This type of cell death is called necroptosis. The key of the necroptosis is the phosphorylation of these three molecules. So these three molecules, the phosphorylation antibody against these three molecules is the important marker of necroptosis. So necroptosis is caspase independent. The cell broken result to the release of intracellular protein. The intracellular protein would be released in the extracellular site. In the extracellular site, this protein acts as a danger signal. The danger signal induce immune response, that is inflammation. So necroptosis induce inflammation. The next question is whether necroptosis has anti-cancer possibility. Here show you the compound and the agent used for cancer therapy. This compound also activates necroptosis. The RIP1, RIP3, or MLKL are activated by this compound and agent. That indicates that targeting necroptosis may be useful for anti-cancer therapy. To demonstrate that, more studies are required. Besides induce inflammation, the danger signal also induce another type of cell death, that is ferroptosis. Usually, ferroptosis is activated by pathogen, follows inflammation formation, and eventually result to cell broken, that result to cell death. Different pathogens, such as bacteria or danger signal or viral DNA or toxin, are sensed by different sensors. The most popular sensor studies is the NLRP3, because NLRP3 is responsible to sense the danger signal. The sensor the sensor complex with pro-caspase-1 and SC to form an inflammasome. The inflammasome induces the activation of caspase-1. The caspase-1 cleave the proform of IL-1 beta and IL-18, then trigger the maturation of IL-1 beta and IL-18. Both IL-1 beta and IL-18 are inflammatory cytokine. Please keep in mind that they are inflammatory cytokine. The caspase-1 also cleave the gasdermin D, GSDMD, gasdermin D. The cleaved gasdermin D has pore-forming ability, so they form a pore in the plasma membrane and result to cell broken, so the cell die. This type of cell death is called ferroptosis. Also, the inflammatory cytokine also release through this pore to the extracellular side. In the extracellular side, it as an inflammatory cytokine induce the inflammation response. So ferroptosis is also induce inflammation. Here I would like to show you Ferroptosis mini-film. After watching this mini-film, you may have a clear idea about the ferroptosis. Let's watch the mini-film.
The neighbor cell is infected.
What's the pathogen?
It is a flagellated bacterium.
Let me check the Guide of Pathogen Defense Strategy. It's an LRC4. Active the LRC4 inflammasome immediately.
Macrophage and neutrophil are here.
You are safe now.
So the next question is whether Ferroptosis has anti-tumor, anti-cancer possibility. Please recall that the immune cells release granzyme to activate Caspase 3 and induce apoptosis. That is cytotoxicity. This year, two groups reported that granzyme not only induce apoptosis but also induce ferroptosis. The group in Harvard Medical School reported that granzyme B cleaved gasdermin E, which form a pore in the plasma membrane and result to ferroptosis. Another group in China reported that gasdermin A, granzyme A cleaved gasdermin B, which then form a pore in the plasma membrane and result to ferroptosis. The result shows that granzyme not only induce apoptosis but also induce ferroptosis. This result indicates that targeting ferroptosis may be helpful to enhance the activity of anti-tumor immunity. Then let's talk about ferroptosis. The characteristic of ferroptosis is lipid peroxidation, which results to plasma membrane broken and eventually results to cell death. The accumulation of ferrous iron, the overaccumulation of ferrous iron have phantom reaction with lipids and generate a lot of lipid ROS. The lipid ROS result to lipid peroxidation, which broke the structure of plasma membrane and result to cell death. This type of cell death is called ferroptosis because ferrous iron is important in this process. In cells, the ferroptosis can be prevented by GPS4. The GPS4 is an enzyme This enzyme uses glutathione to reduce lipid ROS. Lipid ROS reduced, so the lipid peroxidation is prevented. Then the following ferroptosis is prevented. The glutathione is synthesized by cysteine, which is imported from extracellular site to intracellular site by cystine SC. So the importing of cysteine for the generation of glutathione, then the glutathione can be used to reduce the lipid ROS and prevent ferroptosis. So the hallmark of ferroptosis is lipid peroxidation, which can be detected by 4-HNE or MDA antibody. Also, I would like to show you a ferroptosis minifilm. After watching this minifilm, you could have a clear idea about the ferroptosis. Let's watch the minifilm.
Do you want to go inside?
Yes.
Follow me. It's time to join the lipid bilayer of plasma membrane. You have lipid peroxide. That will disrupt the plasma membrane. Let me remove the peroxide before you join the lipid bilayer. Give me some glutathione. Too much peroxides to remove. The plasma membrane is damaging. Oh no.
The same question is whether ferroptosis has anti-cancer possibility. The answer is yes. A report in Science Journal this year, the group in Columbia University reported that cysteine depletion induced pancreatic tumor ferroptosis. Pancreatic cancer cells has a high level of ROS, but the tumor cell would not die because the tumor cell managed to avoid ferroptosis by importing cysteine. The tumor cell imports many cysteine to generate glutathione that can be used to reduce lipid ROS and prevent ferroptosis. So the group use a drug, cysteinase. It's a drug inhibits cysteine importing. By this drug, by treatment this drug, the cysteine importing is inhibited. So the no glutathione synthesized. So the GPS4 cannot reduce the lipid ROS. Then the overaccumulation of lipid ROS results to lipid peroxidation and eventually cause to cell death, the ferroptosis. So targeting ferroptosis can be used for the anti-cancer treatment. Let me summarize the apoptosis. Apoptosis works through Caspase cascade. There are intrinsic and extrinsic pathways. The immune cells mediate cytotoxicity to eliminate tumor-infected or other harmful cells. To facilitate the research of apoptosis, arigo offers apoptosis marker antibody duo, in which contains caspase-free and PRP antibodies. Both are popular apoptosis marker antibodies. For intrinsic pathway studies, arigo would recommended the Bax to BCL2 ratio antibody duo, in which contains Bax and BCL2 antibodies. The high ratio of Bax to BCL2 means apoptosis. For cytotoxicity research, arigo offers granzyme B and perforin antibodies to detect the expression level of granzyme B and perforin in the tumor tissue. The high level means high cytotoxicity. High cytotoxicity means a high ability to kill the tumor cells. Besides, arigo also offers granzyme B ELISA kits and perforin ELISA kit to detect the expression level of these two proteins in serum and plasma. The necroptosis is a backup of apoptosis because it is caspase independent. Necroptosis induces inflammation. For necroptosis research, arigo recommends our necroptosis marker antibodies that includes phospho-RIP1 antibody, phospho-RIP3, and phospho-MLKL antibodies. All these three antibodies are essential for necroptosis research. They are necroptosis marker. Ferroptosis usually is pathogen dependent, and the inflammasome formation. Ferroptosis also induces inflammation. To facilitate the research of ferroptosis, arigo offers ferroptosis antibody panel, in which contains the antibodies required for ferroptosis research. Besides, arigo also offers NLRP3 inflammasome antibody panel and the MLRC4 inflammasome antibody panel. Both antibody panels are useful for the inflammasome research. The hallmark of ferroptosis is lipid peroxidation. To facilitate ferroptosis research, arigo offers ferroptosis oxytosis antibody panel. This panel comprises the key antibody for ferroptosis research. I would like to highlight the 4-HNE antibody. This antibody is used to detect the lipid peroxidation. That is the hallmark of ferroptosis. The oxytosis is a cell death type in neuron cells. Both oxytosis and ferroptosis share similar pathway. So this antibody not only useful for the study of ferroptosis in tumor cell, cancer cell, but also useful for the study of oxytosis in neuron cells. That's the ferroptosis antibody panel. Besides the antibody panel and the antibody duo, arigo also provides a programmed cell death poster. In this poster, the pathways of apoptosis, necroptosis, ferroptosis, and ferroptosis all are in this poster. And also, arigo offers promotion. The customer gets a free multiplex secondary antibody panel with any order of arigo's duo and panel product. The multiplex secondary antibody panel comprises 5 useful secondary antibodies: anti-chicken, anti-rat, anti-rabbit, anti-mouse, anti-rabbit, and anti-goat secondary antibodies. That's the update about the targeting cell death and the product arigo can offer to facilitate related research. Thank you for your attention. Any question about cell death or about arigo's product? Any question? Any question? Targeting cell death is a trend for cancer therapy because the researchers find that targeting apoptosis only targeting apoptosis is not enough to kill the cancer cells efficiently. So many people want to combine the multiple cell death targeting to increase the effect of anti-cancer treatment. That's why targeting cell death attract attention. Any question about today's webinar? Any question? If no question, I think we will end today. Thank you for your attention again. If no more question, I'll say goodbye. Any question? No more? Oh, thank you and goodbye.